Hello and welcome back to Animal Sciences 142. In this week's lectures, we're going to focus on the anatomy and physiology of the cell. Now this lecture is actually divided into three parts. In the first part, we're going to take a look at the function of various cellular organelles. In the second part, we're going to focus on cellular reproduction and mitosis. And in the third part, we're going to focus on membrane transport processes such as osmosis and diffusion. So we're going to start out talking a little bit about the properties of cells. If you remember back to the first lecture, we said that cells were the smallest unit of life that still demonstrate all the properties of life. That is, they have a metabolism, they're able to maintain homeostasis, and they're also able to reproduce. Now you should also remember that cells are usually very, very small, somewhere between 10 to 50 microns in diameter. If you forgot how small a micron is, remember that micro means 1,000, and so 1 micron is 1/1000th of a millimeter. So most cells are really, really small, pretty much microscopic. And the reason that cells are small is that small cells have a proportionally larger surface area through which they can absorb substances they need. That is, they can move things across the membrane a lot easier if they have a smaller internal volume to a larger external surface area. Another reason that cells are small is that a single nucleus can control the metabolic activity of a small cell much easier than it can a large one. So we can classify cells into one of two types. Cells are either prokaryotic or eukaryotic. Now, prokaryotic cells are cells that don't have a membrane-bound nucleus and also lack membrane-bound organelles. Now, in most cases, prokaryotes are small, on the order of 100 nanometers to up to 5 microns, and they're exclusively unicellular. And the primary example we're going to give you for prokaryotic cells are bacteria. So things such as the leptospirosis bacteria, salmonella bacteria, they're all prokaryotes. On the other hand, animal cells are eukaryotic. That is, they have a true nucleus and they also have membrane-bound organelles such as endoplasmic reticulum, mitochondria, and so forth. Okay, now let's take a look at some common prokaryotes we might encounter in the veterinary clinic. Examples of pathogenic or disease-causing bacteria include things like leptospirosis. Now, leptospirosis is a disease caused by a spirochete bacterium. Now, spirochete just means it has a spiral corkscrew shape, and this is a bacteria that's transmitted through feed or water contaminated with infected urine. So usually things like mice and pigs and things like that can spread leptospirosis in the water supply. So if your dog ingests the bacteria, it may come down with a leptospirosis disease, which can cause a lot of problems. It can cause liver damage and kidney failure, among others. Another type of pathogenic bacteria is Salmonella. Now, Salmonella is a rod-shaped bacterium that is commonly found in poultry, and that's the reason we really have to cook our chicken really well, but it's also part of the normal skin flora of reptiles. And this is the reason why we don't necessarily want small children handling turtles and things like that, because they can accidentally ingest the Salmonella bacteria. Now, once it enters the human body, it can cause gastrointestinal distress and enteritis, and so it's not a very pleasant infection. And finally, kennel cough or tracheobronchitis is a disease which in part is caused by a bacteria called Bordetella bronchicipta that is found in dogs. And so I'm sure you're aware, if you take a dog in to get boarded or someplace like that, oftentimes they'll require your dog be vaccinated for Bordetella in order to prevent infection of other dogs in the boarding area. Now, at the bottom of the slide, I pose a question to you. That is, which of these diseases above are zoonotic? If you remember back to the first lecture, we said that zoonotic diseases are diseases which can be transmitted between animals and humans. And so which of these three diseases are zoonotic? If you said leptospirosis and salmonella, you're correct. Both of these are diseases which can be transmitted between animals and humans, whereas Bordetella bronchicipta cannot be transmitted to humans, so it is not a zoonotic infection. Okay, now that we've talked about the prokaryotes, let's spend some time talking about eukaryotic cells. Now remember that eukaryotic cells have clear membrane-bound organelles and a nucleus, and eukaryotes can be either unicellular or they can be multicellular. Now, of course, animals are multicellular. They're actually made up of trillions of cells in most cases, but we also have some unicellular eukaryotes, which are collectively known as protists. These include things such as the amoeba and the paramecium. Now, some of these are disease-causing and some of them are quite benign. So an example of a pathogenic or disease-causing single-celled eukaryote is the coccidian parasite Toxoplasma gondii. Toxoplasma gondii is a unicellular eukaryotic parasite, and cats are the definitive host for this parasite, meaning they're the host in which reproduction can occur. However, it's important to realize that almost any warm-blooded vertebrate can be infected by this parasite. In fact, recently there was accounts here in Hawaii of 2 monk seal deaths being caused by Toxoplasma gondii. Now, the eggs are usually released in the cat's feces, and the way that humans can become infected is by changing the litter box. And normally this infection doesn't cause much in the way of problems, but it can cause fetal defects in pregnant women, and for that reason, pregnant women should not be changing any of the litter boxes. In the past few years, we've also come to understand that chronic toxoplasma infections can also affect mental disorders. So people that, for example, have a predisposition to developing schizophrenia or something like that, if they're toxoplasma positive, they might have a slightly greater chance of developing the disease. They've even found that people that are toxoplasma positive may actually take risks more often than people that do not have a toxoplasma infection. So far we've talked about diseases that are caused by either prokaryotic or eukaryotic cells, but it's important to realize that some diseases are caused by non-cellular critters, such as viruses and prions. Now, a virus really isn't a living organism. At least most people don't classify it as such. It's basically proteins that are encapsulating either DNA or RNA. And usually the virus doesn't have any metabolism or signs of life unless it's infected a mammalian cell or other type of cell. And it's only within the cells of a host organism that viruses can replicate. And so there's lots of different pathogenic diseases that are caused by viruses. In the veterinary field, we have parvo, which causes gastroenteritis and can be a leading cause of mortality in young unvaccinated puppies. Other examples of viral diseases include distemper and feline leukemia virus. Now, it's important to realize that some of these viral diseases can be zoonotic. For example, rabies is a zoonotic disease caused by the rabies virus. This can be easily transmitted in the saliva of animals to people, and once people are infected, if they develop the symptoms or signs of the disease, mortality rate is nearly 100%. For that reason, it's important that people in rabies areas get the prophylaxis and also that they get prompt medical attention if they're bitten by an animal suspected of having rabies. Another type of non-cellular disease-causing agent is something called a prion. A prion is basically an abnormally folded protein which is found within the nervous tissue of some animals, and somehow it propagates itself within the nervous tissue and causes bovine spongiform encephalopathy in cattle. This is otherwise known as mad cow disease. Now, it's important because these prions are found within the nervous tissue, but can also be found in small amounts within blood and the meat tissue as well. And they are zoonotic. They can be spread to humans, and most importantly, the BSE agent isn't inactivated by cooking at normal temperatures. So there have been cases in Britain where hundreds of people have fallen ill and died by contracting BSE through tainted meat. Okay, so now that we've had an overview of the different types of cells and also non-cellular organisms and disease agents, we're going to go back and focus on eukaryotic cells because those are the cells that we find within animals. Now remember, eukaryotic cells have a nucleus and membrane-bound organelles. Now surrounding the cell is something called the cell membrane or plasma membrane. Now the plasma membrane is important because it acts as a flexible, elastic barrier between the inner cytoplasm and outside environment. And so it's important because it governs the movement of molecules into and out of the cell. I like to think of it as being a bouncer at a very trendy bar. They might let one person through, but they may not let you through. Now the plasma membrane is composed primarily by 3 different types of molecules: phospholipids, cholesterol, and proteins. Now the majority of the cell membrane is actually composed of phospholipids. Now remember that phospholipids are a weird lipid in that they have hydrophilic heads and hydrophobic tails. And these lipids are oriented in a bilayer. And basically they allow small nonpolar molecules to cross as well as other lipids, because remember, one lipid can easily dissolve into another lipid. On the other hand, ions and water-soluble molecules usually cannot cross the phospholipid bilayer because they're not lipid-soluble. Instead, they have to cross through membrane proteins. Now, membrane proteins can either be integral proteins, which span the entire width of the membrane, or they can be peripheral proteins. It's the integral proteins which basically act as tunnels for these large or polar molecules to enter and exit through the cell membrane, and it allows them to bypass the phospholipids in the cell membrane. On the other hand, the peripheral proteins are found both inside and outside the cell membrane and may be important for catalyzing enzymatic reactions and also allowing for cell-to-cell recognition. Now, while we're on the subject of the cell membrane, we need to make mention of two cellular projections that we occasionally find on animal cells, and these include cilia and flagella. Now, cilia and flagella are both whip-like projections of the cell, but cilia are much smaller and more numerous. For example, we find cilia on a paramecium, and this helps to propel the paramecium throughout an aqueous environment. Within animals, we find it within the respiratory tract. The cilia here are not responsible for moving the animal per se, but they do move waves of mucus throughout the respiratory tract. Remember that mucus is there to trap dirt and debris, but it only has a relatively short lifespan. Eventually it's going to dry out, and so we need to get rid of it. And so this mucus is propelled up the respiratory tract by the ciliary motion, and eventually the animal either coughs it up or swallows it. A second type of cellular projection is called a flagellum. Flagellum is singular and flagella are plural. Flagella are very long whip-like appendages that help to propel the cell. Usually they only have one flagellum, whereas another cell might have several cilia. So the one place within mammals that we'll find a flagellum is on the sperm cell. The sperm cell is propelled through the female reproductive tract by this back and forth motion of the flagellum. So now that we've talked about the structures on the outside of the cell, we're going to turn our attention to the inside of the cell. Before we do so, I want to make mention of something called the cytoskeleton. The cytoskeleton is basically a three-dimensional network of protein fibers and cables within the cell that give the cell its unique shape, whether it's columnar or cuboidal or squamous, and it also acts as a monorail system that guides vesicle transport within the cells. So we're not going to spend much time in lecture talking about the cytoskeleton, but please do read about it in your textbook. So everything that is inside the cell membrane is considered part of the cytoplasm. The cytoplasm includes the cellular jelly or cytosol, and it also includes the organelles such as the endoplasmic reticulum, the mitochondria, etc. So organelles are membrane-bound structures within the cytoplasm that possess specialized cellular functions. The membranes of these organelles are similar in composition to the cell membrane. That is, they tend to be made of a layer of phospholipids. Okay, the first of our organelles is the mitochondrion. The mitochondrion is important because it's the powerhouse of the cell. It allows the cell to convert molecules such as proteins, carbohydrates, and fats into ATP. Now remember that ATP, or adenosine triphosphate, was the primary energy source that we use on a cellular level. Cells use ATP, and they get that ATP by metabolizing larger food molecules. And so it's important to realize that very metabolically active cells, such as skeletal muscle and cardiac muscle, have lots of mitochondria, whereas cells that aren't as metabolically active don't have near as many. Okay, our next organelles are very small but still very important because these are the ribosomes. The primary job of the ribosomes is to manufacture proteins. Now, ribosomes can be found in one of two different places in the cell. A lot of them are floating free within the cytosol, whereas others are found connected to other organelles. An example of an organelle that may have ribosomes attached to it is the endoplasmic reticulum. Now, the endoplasmic reticulum is composed of a series of flattened tubes stacked on one another and bent into a crescent shape. Now, the walls of the endoplasmic reticulum are composed of a single layer of phospholipids and are continuous with the membrane of the nucleus and Golgi apparatus. Now, there are two different types of endoplasmic reticulum, or ER. Now, rough ER contains ribosomes, and so its primary function is the manufacture of proteins that are going to be used or exported outside of the cell. On the other hand, smooth ER does not have ribosomes. Instead, its job is to manufacture lipids and also act in detoxification and metabolism of drugs. Our next organelle is the Golgi apparatus. The Golgi apparatus is basically the FedEx of the cell. It's responsible for refining, packaging, and shipping substances that are going to be exported from the cell into the extracellular space. For example, proteins that are made on the rough endoplasmic reticulum will travel to the Golgi, be modified, and then exported from the cell via exocytotic vesicles. We'll talk more about protein packaging and shipping when we go through the processes of transcription and translation in a few more slides. So most of the organelles we've looked at so far are responsible for making or secreting something. On the other hand, the lysosomes are responsible for breaking things down. They're basically the cellular stomachs. They're responsible for breaking down molecules into smaller molecules, and they're also responsible for recycling worn out old organelles. For example, if you have a mitochondria that's broken down, no longer working, the lysosome can help to break that organelle down and recycle its components so that it can be reused. Another organelle similar in shape and size to the lysosomes are the peroxisomes. Now, peroxisomes are responsible mainly for detoxification. They contain powerful enzymes such as peroxidases and catalases that help to detoxify things such as formaldehyde and alcohol and break it down into less hazardous substances. As you might guess, peroxisomes are most numerous within cells in organs that do a lot of filtering and also detoxification. For example, the kidneys and also the liver. Our next organelle is the centrioles. The centrioles are small, hollow cylinders composed primarily of microtubules. Now, they're usually only going to be visible when the cell is undergoing mitotic cell division. During these cases, the centrioles help to organize the microtubule spindles, which will later invade the nucleus and grab a hold of the chromosomes in preparation for separating them during mitosis. We'll talk more about the centrioles during part 2 of this lecture, which focuses on mitosis and cell division. Now, last but not least, our most important organelle is the nucleus. The nucleus is the cell's control center in that it contains most of the DNA within the cell, and we'll talk about why DNA is really important here in just a second. Now, the anatomy of the nucleus includes an outer nuclear envelope. This envelope you see has lots of pores in it, which is important because this allows for the passage of molecules such as RNA. The nucleoplasm is basically the intercellular jelly that's found within the nucleus. And finally, the nucleolus is the darkened region within the nucleus that is responsible for the manufacture of ribosomes. Now, we said in the last slide that one of the reasons the nucleus was so important is that it contained the majority of the cell's DNA. You probably already know that DNA, or deoxyribonucleic acid, is important because it is a genetic blueprint molecule. It contains all the information to make an entire new cell, indeed an entire new organism. On the other hand, it also contains a lot of instructions for day-to-day activities such as protein manufacture. So DNA is really important. Now, in most cases, the DNA within the cell is not visible using a microscope. That's because it's unwound into this diffuse mass called chromatin. On the other hand, as a cell gets ready to divide or go through the process of mitosis, the DNA molecule will wind around proteins called histones to form visible structures that are called chromosomes. We'll talk more about chromosomes in the second part of this lecture on cell reproduction and division. Okay, now that we know a little bit about the functions of the various organelles within the cell, we're going to spend the last few slides of the lecture going over protein synthesis. I said once before that a lot of the cell's job on a day-to-day basis is to manufacture protein, and that the instructions for manufacturing these proteins are found within our DNA molecules within our nucleus. I want you to think of the DNA molecule as a giant cookbook with lots and lots of recipes for different types of proteins. Now, the process of making proteins is quite complex and probably beyond a veterinary technician class. However, you should know that it's divided into two parts, transcription and translation. Now, the first step in the process is called transcription. Transcription occurs in the nucleus, and it's the process by which genetic information is copied from our DNA molecule into an RNA molecule. Now remember, the DNA molecule is a cookbook. It's got lots and lots of recipes, and we only want one recipe at a time. So we simply copy down a single recipe into a much smaller molecule called messenger RNA. That messenger RNA molecule is a single-stranded molecule that can then easily leave the nucleus through the pores in the nuclear membrane and travel to the rough endoplasmic reticulum or to the unbound ribosomes within the cytosol. Once the mRNA transcript reaches the ribosomes, we'll then go through the process of translation. That is, the ribosome here will sort of act as a chef, it will read all the information on that recipe or mRNA template, and it will use that information to guide the assembly of amino acids into individual proteins. For example, it will tell the ribosome that it needs to attach a leucine to an isoleucine to a valine, etc. These are all different amino acids, and it's the sequence of the amino acids which determines the primary structure of a polypeptide or protein. Now remember that if this protein is being made by a free ribosome, it's going to be used within the cell. On the other hand, if it's made by a bound ribosome, that is one attached to the rough endoplasmic reticulum, that this protein is eventually going to be exported outside of the cell. But before that happens, we need to modify it a bit. For example, that protein will bud off the face of the rough ER and then travel to the Golgi apparatus. Now within the Golgi apparatus, it will be modified and tagged for shipping to certain destinations. It will then leave the opposite face of the Golgi apparatus and travel to the plasma membrane or cell membrane via something called exocytotic vesicles. And finally, these vesicles will merge with the cell membrane and basically exude the contents into the extracellular spaces. For example, insulin is an amino acid or protein hormone which is made within the cell but later exported outside of the cell.